when you're talking about eukaryotic transcriptional regulation, you have to know, again, that you have two forms of transcription. You have basal levels, and you can also have increased transcription. You can activate it, and you can attenuate it. You can modulate the expression above or below the basal levels. And there are transcription factors that bind to segments in the DNAs, that they're not in the promoters. Those are in enhancer sequences. Usually the enhancer sequences are way upstream of the promoters. Now remember which side is upstream? Is it before or after the gene codes? Yes, how many for before? How many for after? Okay, upstream. Everybody forgot about the picture of the guy sleeping in the water and going down the stairs? Yes, the promoter is the water puddle. So upstream, up the stairs. Right? The promoter is way before upstream, and then the gene is downstream. So if I say the enhancers are usually upstream, they come way before the promoters. Okay? So we're talking about enhancers, will interact with the promoter region, and it can either activate or delay the process of transcription. Now, this is another very important thing to know. What are the levels of regulation in an eukaryotic cell? You have to know how can you regulate eukaryotic gene expression. Where can you regulate it? First thing, you look at the cell, and what is it you see? Yes, when you look at the cell, eukaryotic cell, what is the first thing you see? The nucleus, yes. So we have a nucleus here. So you have your DNA right there. And you say, okay, let me look at the DNA. Structure of the DNA. If I keep it all coiled up, I'm going to inhibit gene expression because there is no way that the promoter and the enhancer sequence, whatever those are promoting or binding on the RNA polymerase, there's no way they're going to reach that DNA if it's all tied up. So that's one level of expression, one level of control of gene expression. Then, okay, let's say you pass that level. You pass by the first stage. You have a relaxed DNA. Now you're going to transcribe it. How, again, can you control this? You can control by, once you have your pre-RNA here, you can control by how this pre-RNA is going to be processed. You have to do splicing, you have to put the cap, you have to put the tail. That's another level of control. If you don't cap and don't put the tail, and then this RNA just degrades before it becomes a processed RNA, this one is going to be gone. You don't have your gene expression, right? If you don't process it, then no expression, another level of control. Let's say you pass the second level. We're giving it a cup of tea, pass the second level. Now you have a processed messenger RNA ready to come out of the nucleus. We're missing on a step here that is very important, and I definitely want you to know this step. I will add when I'm doing the video on this. What is missing here between processed RNA and translation? Okay, come on guys, this is my favorite protein, my favorite complex. Yes, they look like a basket, right? Nuclear pore complex. You have to remember those. They're right at the membrane in the nucleus. They control what comes in and what goes out. Very important level of gene expression. I tell you, it's very important. All the viruses know about it, especially the very pathogenic viruses. Ebola, HIV, influenza, they all know about that. This is how they get us so sick. Control of gene expression right at the gate. Messenger RNA can't come out, you can't make protein. Sorry. Okay, very important level of gene expression control here at the nuclear pore. Now, okay, let's say this messenger RNA passed the nuclear pore, now it's in the cytoplasm. It's going to make the protein. Once it's in the cytoplasm and it goes through the translation, there's another level of protein expression here, control, because you can have post-translation modifications of a protein. If your protein, some of the proteins do not get post-translational modifications and proper folding, chaperone foldings, and proper addition of side molecules and groups that get this protein to work. If that doesn't happen, your protein doesn't work. it doesn't really matter that you went through all of this control to get in there and then your protein doesn't work. Another control of gene expression. And so eukaryotes have a lot more room to control the gene expression. So you have here some of the eukaryotic controls. They're regulating the transcription. Okay, so those are transcription factors. Remember that you can have transcription factors that are present in every cell. Those are the housekeeping genes. And you also have transcription factors that are present only in certain types of cells. Those are specific gene expressions for certain types of cells. You also have histone acetylases. What are those doing? Modifying histones.
Take them apart,
Right.
separate them from the
DNA. Yes, they are going to relax the grip of the histones on the DNA. You remember what histones look like? Yes, octamers, those little balls, right? Little balls in the forms or little things. Okay. I just listen to their names. You know, these stones, they have to be this form of complex. Okay. Now, histone acetylases, what does it do? Modifies those little balls. No too much grip on the DNA. Level of control right there. Now, okay, transcription factors. You can reduce transcription. You can enhance transcription. Now, you also have here, is it shown here? No. Okay, let's go to the next one. DNA methylation. What is it that methylation does to your DNA? Inhibits gene expression. Methylation is a form of shutdown of gene expression. Methylation happens when you have cytosines here that can receive these methyl groups from methyltransferases. Okay, so there's your cytosine right there, and then you stick a methyl group right there, and you stop the expression of that gene. The more methylation you have, the less gene expression you have. Now, the control, methylation control. We have here these methyltransferases, the methylated bases, but you do have this transfer of methyl groups to cytosines. That part you have to remember. And they will block the chromatin remodeling. They will keep everything stuck together, and also block the histone acetylases. And those can also recruit the deacetylases. Now, deacetylases are avoiding the acetylation of the histones, and if anything gets acetylated there, those deacetylases are going to make sure that it gets deacetylated right away. So if you try, if one of those hats getting there and acetylates, with the methyl groups attracting the acetylases, that process is going to be reversed right away. Now, again, methylation here usually functions to block transcription. Now, again, it's tissue specific. Sometimes you need transcription in one tissue and not in the other. So this type of methylation and just this type of blocking of gene expression will happen during embryonic development when you are differentiating the cells. Remember, every cell has your whole genome. How do you control what genes are expressed in each cell? Well, we have to have some type of transcription control. Methylation is one of them. For example, here, if you want brain-specific proteins to be expressed, you're going to block other proteins. Then if you want liver proteins to be expressed, you're going to block like brain-expressing proteins, because you don't want brain-expressing proteins to be active in your liver. So it has to be tissue-specific. The methylation has to be very, very specific. Now here is an interesting thing. When you have the cloning, okay, the cloning your pets type of problem, what happens here? Well, we know that control of gene expression has to do with the methylation, and this is a type of gene expression that is epigenetics. Remember what is epigenetics? Anything but sequence of DNA. Anything that is acting on DNA and is not directly altering the sequence of the bases. That is epigenetics. And when you are making a clone of an animal, when you are cloning something from a somatic cell, you get a somatic cell, let's say from the skin, and you're going to get this empty egg. You take the DNA out of the empty egg, and you put the DNA from your somatic cell into that nucleus of the empty nucleus of that egg. Theoretically, when you do that, all of the methylation that was present on the somatic cell, on your skin cell, all of the methylation is supposed to disappear. And you are supposed to get a total reset of the entire genome that you put inside that egg, that empty egg. Now, do you think that happens? No, this would be an ideal situation in a perfect world, which is not quite the world we live in. So, in the world that we live in, when you're doing cloning of animals, you get a somatic cell, and you do not get a total demethylation. Some of it still sticks in there. So what is the consequence of that? You are going to have problems with the differentiation of the cells in that embryo, because some of the genes are already going to be deactivated, and you cannot possibly get a proper differentiation and proper gene expression on every single cell of this clone, because a total reset of the genome does not happen. Okay, so you're going to have problems. And one of the problems is that most of the clones will die very young. So this is already the second problem that we found about the cloning your pet problem. The first one, you remember what it was? The telomeres. Telomeres are shorter, so that means the cell is older. That means the pup is born already 16 years old, if it is a dog, for example. The other problem is methylation. You get a pup that is very old, and you get a pup that it has methylation before it developed embryonically. So it is even a bigger problem now. And then you think these people spend $50,000 cloning their pets. And we know also the methylation is depending on the environment. We know that the methylation is also influenced by the environment. So depending on what the environment situation is, you're getting some epigenetics here or there, so the results are going to be different, even if you're cloning this animal from a somatic cell from scratch. And an example here is with this cat. I think this is the clone, this is the mother, sort of. What happened is that they wanted to get the cat cloned, but it did not come out exactly the same as the mother. It's a different cat. It has different hair, coat pigmentation, and totally different behavior. Now, how did that happen? Well, one explanation is because of the different methylation patterns that was expressed on the DNA. Another important thing to remember on gene expression is what we did again here on epigenetics of imprinting. I remember the case of, what are those two diseases, those two syndromes? Prader-Willis and Angelman syndrome. Those are from the same chromosome, but because of the nature, because of the origin of the parent where the chromosomes came from, the genes are going to be expressed differently. Why? Because you have different patterns of methylation if that chromosome came from the man or from the woman.